In this video, we will discuss about the disease Bacillus anthrax. This has several synonyms such as splenic fever, wool sorter's disease, and malignant carbuncle. Splenic fever, this is because of the gross lesion in the spleen in which the enlarged spleen will happen, that is known as splenomegaly. And wool sorters, it is because of this is mainly found in the human beings, those who work in the sortering of wool. And malignant carbuncle, this is the type of skin disease where large rashes on the skin will occur. This anthrax is acute infectious septicemic disease, which means the mortality rate is very high, that is sudden death will happen, that is acute. And infectious, that is caused by the Bacillus anthracis, and the Bacillus anthracis will produce the toxins in the blood. That's why it is called as septicemic disease. This is commonly affecting the herbivorous animals such as cattle, sheep, pig, characterized by high fever, sudden death, and enlarged spleen. The Bacillus anthracis is gram-positive, capsulated spore-forming rod. This is caused by the Bacillus anthracis. Let us come to the characteristics of the Bacillus anthracis. Etiology of the anthrax is Bacillus anthracis is pathogenic strain causing anthrax in both human beings as well as animals. It is non-motile, capsulated, aerobic, spore-forming, gram-positive organism which appears as straight, large rod-shaped or small short chain. The primary host is ruminants or herbivorous animals, and the human act as accidental host. This disease has zoonotic importance. Epidemiology of anthrax in animal and human host. The spores which are present in the environment as in soil, they are heat resistant. The spore may enter the cattle while grazing, which leads to the death. And sometimes the spore will contaminate the hide of the herbivore. Then in case of accidental host in human being, the mode of infection is via contact with the contaminated animal products, hides, wool, hair, and or by ingestion of the contaminated food product or by inhalation of the contaminated dust. That's why it is called as wool sorter's disease, in which the human, those who are working in the wool sorters industry, the high factor is that in which the disease will occur. Now let us come to the pathogenesis. It is either because of bacillus or because of spore. The transmission is mainly through ingestion or inhalation. If pathogenesis because of bacilli, then it is called as bacilliform. If pathogenesis is because of spore, then that is called as sporeform. The bacilli will enter the host via inhalation or via ingestion. If the bacilli is ingested, then it will reach the pharynx, then it will proliferate into the lymph node, then from the lymph node it will goes to the other organs where it will produce toxins. If the bacilli is ingested, then it will reach the stomach by esophagus, then where it will destroyed by the gastric juice, it is produced by the parietal cells. Then in case of spore form, it will enter to pharynx, then it will reach the stomach. From stomach it will goes to intestine, where it will penetrate the intestine and enter into the blood circulation, leading to the septicemia. Septicemia is because of capsule and exotoxins. Capsule of the Bacillus anthracis has two main important properties. The one important property is that which is fibrolytic in nature, and another one is antiphagocytic property. The capsule contains glutamic acid, that is poly-D-glutamate. This is the most asked question in any examination. The capsule of Bacillus anthracis contain dash. Just try to remember that is the poly-D-glutamate, which is fibrinolytic property, thereby it prevent the blood clotting, thereby blood not able to clot, which will lead to the flow of the blood via natural orifice. Then in antiphagocytic property, it protect the bacteria by the phagocytic organisms. Now the exotoxins, mainly there are three type of exotoxins are there. They are edema factor, protective factor, lethal factor. Edema factor will ultimately lead into the production of edematous or swelling lesions. This is by undergoing cyclic AMP production, which will lead to the fluid loss from the cell, produce edema. Then protective factor will help in the antiphagocytic property, thereby it will prevent the destruction of the bacilli. Then the lethal factor is responsible for the death. The lethal factor will stimulate macrophage. Macrophage will in turn stimulate production of oxygen free radicals such as TNF and leukotriene 1, which will lead to the vasodilation. Because of vasodilation, the blood circulation will decrease, therefore which ultimately result in shock and death. Death in the anthrax is because of shock and anorexia. Now let us come to the clinical signs and zoonosis. There are mainly two forms, one pre-acute, another one acute form. In case of cattle and sheep, there is sudden death in case of pre-acute form, and there is no any clinical signs such as fever, muscle tremors, dyspnea, congestion of mucous membranes are noticed. Animal collapses and die due to terminal convulsions. After death, the blood will ooze from the nostrils, mouth, anus, sometime from vulva also. In acute form, incubation period of the disease is 1 to 2 weeks. The course of this form is about 48 hours, and there is high fever up to 48 degrees Celsius, severe depression, congestion, hemorrhage of the deep mucous membrane. There is increased heart rate and rapid respiration is observed. Affected cattle with mouth breathing due to oxygen hunger. In case of pregnant animal, it may lead to abortion. In case of lactating animal, decreased milk yield. In case of horse, always horses are affected with the acute infection by ingestion and leads to the septicemia, enteritis and colic. Painful edematous subcutaneous swelling appear in throat over the lower neck, floor of the thorax and abdomen. This is high fever, severe depression, dyspnea due to the swelling of throat or colic due to intestinal irritation. The course of the disease in horse is about 48 to 96 hours. Now let us come to the gross lesions. The postmortem examination of the anthrax is prohibited. This is because of the concern of the spores may contaminate the environment. Then edema and hemorrhages may be seen in any part of the body, particularly serous membrane. The spleen is greatly enlarged and engorged with the dark unclotted blood. Lymph nodes are swollen, edematous, occasionally hemorrhagic. Hemorrhages and swelling may occur in the intestinal tract, liver, and kidney. In sheep and horse, splenomegaly is not majorly noticed. In swine, edema and hemorrhages are seen in the pharynx and cervical lymph nodes. This is the microscopic slide view of the bacilli. Then let us come to the microscopic lesions. The large number of bacilli are demonstrated in the smear or tissue sections. The anthrax rod can be differentiated from the gram stain. The end of the anthrax bacilli are trunculated in nature. Then let us come to the diagnosis. The demonstration of the capsule is by methylene blue McFadden's reaction. Blood smear stained by the Lessman method will reveal characteristic rod-shaped organism with trunculated ends possessing distinct capsule. They may arranged singly or in very short chains. This is the blood smear in which you can clearly observe the Bacillus anthracis. This is all about the anthrax. In next video we will discuss about brucella disease. Thank you.